Hello everyone, I'm Ali Nikakhtar, your host here at DystroBlog, a space dedicated to discussions and awareness about neuromuscular disease. Today, we're going to explore one of the most recent and important papers about the role of dystrophin in muscle regeneration, a study just published on one of the leading scientific platforms. If you're familiar with Duchenne muscular dystrophy, you probably know the name dystrophin. It's the key protein responsible for maintaining the stability of muscle fibers. Without it, muscles gradually weaken and degenerate. But here's the big question. Is dystrophin's role limited only to muscle fiber structure? This paper directly answers the question, and the answer is fascinating. Dystrophin is also essential in muscle stem cell, not just mature fibers. This study was led by Dr. Matthew S. Alexander, a neuroscientist and leading researcher from the University of Alabama at Birmingham in the United States. He has spent years working on gene therapies for muscular disease. Among the others, you'll also see some legendary names. Dr. Louis M. Kunkel from Harvard University and Boston Children's Hospital. Yes, the very scientist who discovered the dystrophin gene back in the 1980s. Just imagine, the man who discovered the dystrophin gene is part of this work. Another outstanding contributors is Dr. Karen Esser, professor of physiology and aging at the University of Florida. She is known for her groundbreaking research on skeletal muscle physiology, circadian biology, and the aging process. Dr. Esser is one of the leading voices in understanding how muscle cells function both in health and disease. With such incredible team of experts, this study is not just technically strong, but also conceptually deep, offering new insights into how dystrophin influences muscles, stem cell biology. If you'd like to learn more about this scientist and hundreds of other who devoted their lives to improving the lives of children with Duchenne muscular dystrophy, check out my collection of summaries at nikilab.net. Now, back to the research. The team asked a very specific question. What happened to muscle regeneration if dystrophin is removed only from muscle stem cells or satellite cells? We already know that in DMD, muscle fibers degrade due to the absence of dystrophin. But is dystrophin also important for the stem cell that rebuild muscle? To explore this, they developed a specific mouse model where dystrophin was removed only from satellite cells, while muscle fibers themselves still expressed the protein. To selectively delete dystrophin in muscle stem cell, the researchers used precise genetic tools called the CRE-LAXP system. Here's how it works. The DMD gene, which codes for dystrophin, was designed in the flexed configuration, meaning it could be deleted if the CRE-recombinase enzyme was present. They used a specific genetic driver called PAX7 CRE-ERT2, which is only active in satellite cells. And to activate the system, they gave the mice a drug called tamoxifen. So when tamoxifen was injected, CRE became active only in satellite cells, deleting dystrophin exclusively in those cells, with no effects on mature muscle fibers or other tissues. To test how these stem cells perform, they injured the tibialis anterior, or TA, muscle using a toxin called cardiotoxin, which destroyed muscle fibers and forces the body to regenerate them. And the results, fascinating. The mice where dystrophin was intact in satellite cells, regeneration occurred normally. But in mice lacking dystrophin in satellite cells, regeneration was severely impaired. New fibers failed to form properly, many nuclei remained centrally located, and the muscle architecture remained disrupted. The researchers observed that without dystrophin, satellite cells had difficulty divided their ability to differentiate into muscle fiber was reduced. Many cells either died or derived into incorrect non-muscle pathways. Immunohistochemistry showed that a large number of these cells were stuck in the G1 phase of the cell cycle and couldn't move forward. Let's pause and dig into what that means. After an injury, muscle stem cells normally wake up, enter the cycle cell, and follow a progression. G1 phase, they prepare to replicate DNA. S phase, they begin DNA synthesis. Later, they choose either keep diving or enter myogenic differentiation, the process of turning into a muscle fiber. But in this study, the cell got stuck in G1. They began the process but couldn't commit the either pathways, division or differentiation. Many were confused, non-functional, or simply stopped progression. Now, why is this important? This means dystrophin isn't just a structural protein. It's essential for guiding satellite cells to make the right decision, whether to divide or differentiate. Without it, the cells call left in limbo. In biological terms, this is called cell cycle arrest. In another part of this study, the researchers used RNA-seq to see which genes were turned on or off when dystrophin was removed. The results? Genes essential for muscle differentiation, like MyoD1 and myogenin, were significantly downregulated. Pathways like Notch, WNT, and TGF-beta were disrupted. Genes involved in self-renewal and stemness were also reduced. These findings suggest that dystrophin helps regulate the balance. To confirm their findings, the researchers also studied satellite cell in culture. Even outside the body, dystrophin-deficient cells failed to grow properly, showed poor differentiation, struggled to fuse with muscle fibers. Single-cell analysis revealed epigenetic and transcriptional disruptions, meaning the very identity of these stem cells was altered. Here's the shocking part. Just removing dystrophin from satellite cells alone was enough to mimic the pathology seen in the full-blown Duchenne models. Even if mature fibers had dystrophin, muscle regeneration still failed when the stem cell lacked it. But this story isn't stop at muscle regeneration. One of the most fascinating parts of this study was how researchers look at the brain, especially Purkinje neurons in the cerebellum. Using a specialized krill line called PCP2 Cre, they deleted dystrophin only in Purkinje cells. And guess what? The mice developed autism-like behaviors. In behavioral tests, the dystrophin-deficient mice showed reduced social interaction. They had impaired spatial memory, shown in tests like Y mass and Morris Water mass. They also perform poorly in novel object recognition. These mirrors once been clinically observed in nearly 1/3 of DMD patients who also showed symptoms on the autism spectrum. Dystrophin isn't just important for muscle stability, it's also crucial for brain, functional, and behavioral. So, therapies that only target muscle fibers may leave the neurological aspects of DMD untreated. In the long run, we may need gene therapies that also reach the brain to full treat DMD. Another compelling part of this paper was comparing the timing of dystrophin loss. In models where dystrophin was missing from development, muscle damage was severe, just like in classic MDX mice. But in conditional models where dystrophin was deleted only in adulthood, the effects were much milder. And here's the twist. Even 2 months after gene deletion, dystrophin protein remains stable in the muscle. That means dystrophin has a long half-life, which is crucial for designing durable gene therapies like AAV, adeno-associated virus, or antisense oligonucleotides that don't require frequent dosing. In the RNA-seq analysis, over 6,200 genes were significantly altered in dystrophin-deficient muscles. Inflammatory genes like NAUS2 were upregulated, and signaling pathways like TGF-beta and ECM remodeling were disrupted, including genes like TGF-B1 and COL4A4. So, dystrophin isn't just a structural protein. It's a molecular orchestrator. Its absence throughout a whole network of signaling pathways far beyond just mechanical instability. So, here are the key takeaways from this amazing research. Dystrophin is essential not just for muscle fiber integrity, but also for stem cell function and brain signaling. Deleting dystrophin in just one cell type, like satellite cells or Purkinje neurons, is enough to cause serious dysfunction, even when the rest of the tissue still has dystrophin. Future treatments for DMD must go beyond muscle fibers and target stem cells, brain, and immune compounds as well. If you found this video insightful, please like, subscribe, and let me know in the comments if you want to learn more about gene therapy approaches for DMD, the role of dystrophin in the brain, or how RNA-seq helps uncover complex disease mechanisms. Thanks for staying with me till then. And don't forget, check out nickylab.net for more scientific summaries, articles, and videos just like this one. And please don't forget, my dream is to see a smile on your lips.